This week we're going to talk about gene expression. So last week we talked about how genes and DNA varies, and this week we'll talk about how it's expressed and what can affect that.
Like always, here's our objectives and vocabulary. The three objectives are explain how multiple alleles can impact one gene, explain how multiple genes can impact one trait, and explain how the environment influences the frequency of alleles in a gene pool. We're using things like sickle cell and malaria and typhoid and cystic fibrosis, and that stuff we'll talk about in lecture. And here's all your vocab. Remember, the vocab is on Quizlet.
So on top of last week talking about gene variations, I also want to talk about remembering the stuff we learned 2 weeks ago, maybe 3 weeks ago, so everything from this year. So let's remember that our DNA, which is all of our heredity information, right? And it can be broken up into genes, which are small pieces that code for a protein, right? The genes are then turned into RNA, right? We make RNA from them. And then our RNA is going to go into a protein when it goes down to the ribosome when we do the translation. And then our protein is going to be our trait that we look at, right? So when we talk about genes being expressed and we talk about traits, that's what we're talking about, right? So our gene is a segment of DNA that provides instructions for making a protein. An allele is different versions of the gene. So remember that you have 2 copies of each gene, right? The homologous chromosomes, you get one from mom, one from dad, right? They come together and you got 2 copies, homologous chromosomes. They have the same genes, but they can carry different information, right? Also remember from all the way to last year that we have dominant alleles and recessive alleles. The dominants are usually a capital letter and they usually code for a protein. Recessive are lowercase letters, and usually they don't code for a protein. So RNA is going to carry the information from the nucleus to the ribosome where we're going to make our proteins. And our proteins is a chain of amino acids linked together in a specific order. Sometimes they're called polypeptides, right? And lastly, a trait is how the protein looks or appears. Usually when we talk about traits, we're talking about physical things like the ability to roll your tongue, your eye color, your hair color, all that kind of stuff. So next, basic gene expression. This is the stuff you guys already know still, so this is a review. So gene expression is the process that genes are going to get turned on and off to make proteins that produce a trait, right? So if you look at this picture on the side, we have our DNA, which is transcribed to your RNA. RNA is translated to a protein, right? That protein is going to function to make the organism work, right? The DNA sequence turns into an amino acid sequence, and the genotype will turn into a phenotype, right? So how your genes are will end up being how you look, right? Remember that there are 2 different alleles. Normally there are 2 different alleles for a gene, right? It's usually that one's dominant, one's recessive. Sometimes they can be codominant, sometimes they can be incomplete dominant. We talked about all that stuff last year, but the way the 2 alleles interact is what's going to produce the trait that we see. Genotype, right, is the allele pairs, right? Homozygous, heterozygous, homozygous dominant, homozygous recessive, or heterozygous. And phenotype is how it's expressed, the physical appearance of the trait. What does it look like when I look at you? Do you have brown eyes or do you have blue eyes? Do you have blonde hair or do you have brown hair, right? Dark skin, light skin, short, tall, all that kind of stuff. So factors that affect gene expression. So what we just did was kind of basic gene expression, right? We go from DNA to RNA, RNA to protein, and protein is your trait. But there are some other factors in life that are going to affect how the genes are expressed, right? So gene expression in individuals and in populations, depending on how you're looking at it, can depend on more than just the actual genotype. Individuals are influenced by environmental factors. And when we do, people who study that are studying epigenetics, and we'll talk about that in a second. Populations, when we look at the population genetics and we look at the genes in a population, populations are influenced by environmental factors, which is having a genetic advantage, and we'll talk about that later, or by genetic factors. And for genetic factors, we're talking about, is it a polygenic trait, or does the trait have multiple alleles, right? We'll get into those as we go through this. So, environmental factors for our individual gene expression. So this is talking about like your own DNA and what can affect it being expressed, right? So think about like twins, right? Twins have the same exact DNA, but they don't always look exactly alike, right? They can lead different lifestyles, they can make different choices in life, which makes them look different. siblings have similar DNA, but they don't always look alike, right? Because of the choices that they make. So what this is called is epigenetics. This is the study of how environmental factors are going to affect how a gene is expressed. So environmental factors can actually affect the chances of genes being turned on or turned off, right? They can actually chemically cause chemicals to go onto the DNA, right? So that we can skip over certain sections so that it doesn't actually read the gene. Right? Examples of environmental factors that could affect how your DNA is being expressed are things like toxins and UV, drugs and chemicals, even things like temperature, right? Or lifestyle choices. So lots of foods that you eat, right, can do this. They can have an epigenetic effect on your genes, right? It can cause, methylation is what's called, but it can cause like little tags to go on your DNA to make it work or not. We'll do an activity of this later on and take a look at it, right? It's really interesting. But when you think of this, I want you to think of like bunnies, right? Or animals that change with seasons, right? So some animals change color with seasons because it helps them blend in, is what we always think, right? But they don't choose like, hey, I'm going to change my color right now, right? It has to do with the length of the day, and that ends up actually affecting how their traits are expressed. Right? We'll talk about it more, and we'll talk about rabbits. All right, take two for this slide. I just messed up. So we're not talking about the individual anymore. We're talking about gene expression in a population. So we're looking at how genes are expressed and how different things affect how they're expressed in the whole population. We're going to start with environmental factors because that's what we just talked about with the individual. So for populations, certain genes, right, can give you a genetic advantage in particular environments, right? And these genes sometimes are genes that cause like genetic disorders regularly, okay? The example, I have two of them. What we're going to look at is sickle cell anemia. Okay, sickle cell anemia, let's see if I can get my pen to write this time, because that's what happened last time and it stopped writing. Sickle cell anemia causes your blood cells to look like this, rather than like that. Okay, so they're supposed to be big and round, biconcave. They kind of look like a snow tube, you know, they look like this, like that. And those carry our oxygen, right? So when they sickle, they like indent and look like a sickle, like Grim Reaper sickle, right? But when they sickle, it makes it so that the malaria can't, it's a protozoa, I believe, that causes malaria. It can't get into the cell, right? It can't get into the cell and live there, so you can't get sick with sickle cell anemia, right? So though sickle cell anemia can kill you if you have both sickle genes, right? If you only get one sickle gene, so if you have this, right, this one, normal, this, heterozygous, this, two littles, this one can kill you because your blood cells all clot together. This one, some of your blood cells look like this, right? And malaria can't actually infect you. This one, where all your blood cells are good, malaria can still affect you. So the sickle cell gene ends up like kind of hanging around the population, and it becomes more prevalent in areas where there's malaria. So if we look at this map shows us all this orange area. That's where you find malaria. But look at that. And this is also where you find sickle cell anemia, right? So the disease becomes prevalent in that area because carrying it is a good thing, and carrying it keeps you healthy. Make sense? There's another one. Next slide. All right, so this one, we're continuing. We're still talking about how certain environments, it's good to have certain genes, right? And it influences how like the genetic frequency is across the whole world, let's say. Cystic fibrosis and typhoid. All right, cystic fibrosis is a disease, right? If you have if you're homozygous recessive, right, it's not so great. If you're heterozygous, normally you'd be normal, you'd never know. If you're homozygous dominant, right, you're fine, okay? What cystic fibrosis does is it causes you to build up mucus in the lining of your respiratory tract, all right? But the problem is you then like cough it up and swallow it, and then it goes through all of your digestive tract, right? It coats like your digestive tract. Now, This cystic fibrosis when you have the full-blown disease can be deadly. Usually, people don't usually live a full life. It's usually limited. I don't know what age. I want to say 40. It could be wrong though. If you have one normal gene and one cystic fibrosis gene, right? This one actually, you make some mucus, but not a ton, right? And the mucus lines the digestive tract, and that is where typhoid bacteria enter your system. So typhoid bacteria, if you drink contaminated water, right? So water that has typhoid in it, if you drink contaminated water, then you can actually ingest the typhoid bacteria, travels through your system, it goes through the lining of your digestive tract and into your system, and it makes you really sick, right? So people in areas where there's lots of typhoid, right, we find that there's lots of cystic fibrosis. The allele to cystic fibrosis is prevalent because having the heterozygous actually is beneficial because the mucus lines the digestive tract and the bacteria can't get in. But there's a chance that if you have a kid with somebody, then you could pass the cystic fibrosis gene, which isn't good. But if you don't have it, maybe you get typhoid, and that's not good either. So it's just a weird thing that happens, and it affects the frequency. So it's like the environment affecting the frequency of where genes are going to show up. Make sense? We'll talk about it more in class. Last kind of gene expression, the way genes are expressed, how it influences things. So the population is also influenced by genetic factors of genes. So this one we've talked about. Multiple alleles you learned last year, okay? That is when you have more than 2 alleles for a gene, like blood typing. That's one you always learn about. That's one they always talk about, right? Blood typing is A, B, and O, right? So 3 alleles, it only 3 alleles, it actually will do 4 phenotypes, right? Everyone only has 2 copies, even though there are 3 in the whole population, okay? So because of that, it creates more variation in the population. So I can be like AA, and I don't know, Miss O'Neill could be AO, right? Or you could be OO, or somebody else could be BO, right? There's lots of different alleles and lots of different genetic combinations that you could have, right? So it creates variation in the population and the expression of it. There's lots of different like phenotypes, right? Bunny's fur does that lots of times. There's like a whole thing with like guinea pig fur and agouti and like the kind of meshed colors. But you did that last year. It just causes more variation. And then polygenetic traits, this one is new, but this is something that you know. All people know this, like kind of innately, right? So what it is that some traits, even though everyone says blue eyes, brown eyes, right? They teach you the basic of genetics, they always start blue eyes, brown eyes. It's not a great one to use because eye color. Does everyone have blue eyes or brown eyes? No, right? Some people have gray, some people have green, some people have hazel, some people have light blue, some people have dark blue, some people have like yellowish in blue, right? There's like a giant variation mixed in, right? That's because eye color is a polygenetic trait, meaning multiple genes influence that single trait. So there's more than one thing that's coming into play here. So when all of these things come into play, it actually makes many possible genotypes and phenotypes, right? So the phenotypes show more of a blend. There's not like a distinct you have it or you don't, right? Like tongue rolling, doing this, you can either do it or you can't. It's a gene, right? So if you don't have it, you can't do it. And if you have the gene to do it, you can do it. Hair color, like skin color, stuff like that doesn't work like that, right? Height, think about if we lined everyone up in class. Everyone isn't 5 feet or 6 feet, right? We have like a variety of heights. So what happens is when we see a polygenetic trait, they show up as a bell curve. When you line everybody up, you got like lots of averages, lots in the middle, a couple on each end, right? This down here, there's a picture over here of human height. Height does that. Some people are really short, some people are really tall, most people in the average, right? Skin color does that, meaning there's lots of them, but skin color varies by environment, right? Let's think about it. Because of UV, the effect of UV, right? That's why people who have lighter skin tend to get skin cancer more often because they don't have as much protection within their skin, even though you can get skin cancer with all colors, right? So different ways that the environment or genetics are going to influence how genes are actually expressed regardless of what traits you have. Last thing I want to mention, right? In this lecture, we went from talking about individuals' genes to talking about a population's genes, and this is the first time we've done this year. So what I want to point out is when genetics is studied at the population level, right, you focus on the variation within the population, not each person, right? And you tend to focus on the allele frequencies, so how often an allele shows up in each population. And when we talk about this, we're moving from our good old friend, this guy. Look at how happy he looks. Gregor Mendel. Yay, right? And we're gonna make a giant jump to this guy over here. Let's make him smile. He looks a little grumpy. That guy, right? That's Charles Darwin. Okay, so we're gonna make a jump from genetics to evolution when we look at actually the allele frequencies and how those change depending on the environment, which we started talking about how genes are expressed and how the environment affects that in this lecture. It's gonna continue for our next unit. So I wanted to show you the differences. So different ways to study genetics. You can study individuals and study genetics, and you can look at their genome. So like the human genome is all the genes that an individual has, right? Or you can look at the population and you can study the gene pool. That's all the genes in the population, okay? You can study a genotype. I can look at your genotype, I can say you're heterozygous or homozygous. Or I can study the population and I can study allele frequencies. And I can say, how often is the dominant allele showing up? How often is the recessive allele showing up? Okay. Lastly, I could study phenotypes. If I look at the individual and I'm just doing genetics, I can look at the phenotypes. And I can look at a family tree and I can focus on phenotypes and look at a pedigree and figure out how traits are being passed and try to figure out genotypes, looking at how traits are being passed in phenotypes. Or if I'm studying population genetics, I look at the distribution of phenotypes and look at things like the bell curve that we were just talking about when I was doing the human height and talking about polygenetic traits. Okay, so this is two different ways to study genetics, depending on if you're looking at the individual level or if you're looking at the population as a whole and seeing what's happening with the genes in the population. So we started this year by looking at the individual level and looking at DNA and working up, right? And we kind of bumped off of last year looking at the individual level of genetics, but we're going to move into looking at the population level and seeing how that changes depending on how our environments change. Make sure you notes in your notebook, write down any questions that you have, and I'll see you Monday. Bye guys.